Hello, Bio 11. Welcome back. I hope you all had a good weekend. Uh, I have a decision to make about how I'm going to uh, give you the exam that is at the end of this week. There's some cons and pros and cons to the two different ways that I want to do it. And one way would be through Blackboard. I'm going to make that decision tonight or tomorrow, so I will let you guys know. All right. I'm hoping to make the decision tonight. In any case, the format of the exam is going to be uh, composed of fill-ins and short answers. So basically, there might be a true or false. The questions are all going to be from the presentations that I've been doing, and they're going to be heavily based on the review questions at the end of the chapters that I review here in these recitations. Also, there will be some questions from within the chapter. So if you guys followed along and you've been filling your books, you should be okay. All right, there's not going to be anything if I haven't spoken that fact or said something about it, then it's not going to be on the test. All right, I don't do that, so I have to make sure that I cover it before I can test you on. All right, so for this class, I like to just keep it simple and keep it to the review questions at the end of the chapters, and add in a few extras from the chapter itself. All right, so I would say 90% of the test is going to be from the review questions, or 95%, and the remaining will be just a few questions from within the chapters. If I emphasized anything, something along those lines, and from the recitations. All right. Okay. Excuse me. All right. Today's chapter is going to be a little bit longer in terms of explanation of the recitation itself, because it's such a thought. It's more of a thought chapter, than anything. Even if we were in lab, we wouldn't be doing too many experiments. We would just be practicing the practicing on the facts of mitosis and meiosis. Okay, so chapter 7 is in your lab book is concerned with mitosis, and chapter 8 is concerned with meiosis. So we're going to start off with mitosis. Okay. So just as a review in terms of the organelles. Oh, and by the way, on the exam, I'm not going to have you identify anything. There won't be any identifications. So I'm not going to have a picture up and ask you to identify this piece or that part. Okay, so it's just going to be straight short answers, one or two word answers, fill-ins, and or a couple of the longer questions from the end of the chapters. Okay, and a few true and false questions. That's it. Maybe, I'm not sure about the true or false. Okay, so let's start off with this slide here. Remember, the nucleus is the location on eukaryotic cells where DNA is located. All right, and the nucleus in eukaryotic cells has a double membrane, an outer and an inner membrane. Like it's a plasma membrane, just like what we discussed 2 labs ago. All right, and of course the nucleus contains the DNA. When you're looking at a cell under the microscope and the cell is not undergoing cell division, then the DNA is going to be in the form of long strands. They're not coiled at all. So you're not going to be able to see it under the microscope. And the nucleus just appears as a smaller darker pigmented organelle within the cell. However, when the cell wishes to undergo cell division, there are going to be some big changes to the nucleus. The nucleus gets larger, the membranes start to dissipate, and the DNA becomes condensed. All right, and it becomes condensed in a special form which we call the chromosomes. All right, when they are condensed, the chromosomes themselves have several parts to them, each part being a chromatid. All right, and when they are in the form of a chromosome, you can see them under the compound microscope. Okay, so let me see. All right, so I'm telling you about how DNA, right, is not visible unless it's undergoing replication. Well, let me be more specific. Unless the cell is undergoing cell replication, cell division. Otherwise, it's just a long strand of the double helix, deoxyribonucleic acid, DNA, right? And it looks sort of like this, right? A double helix. Now, when the double helix wants to coil up and condense into a chromosome, it does so with the use of many, many different types of proteins. All right, and the most famous one, or the most well known, is the histone proteins. All right, so it coils up around that, and it's coiled up nice and tight. The equivalent of several miles of length is coiled up into a microscopic structure that we call the chromosome. All right, so here I'm pointing at a chromatid. Here I'm pointing at a chromatid. All right. Now this is where it gets a little bit confusing. All right. The chromosome, this whole thing is a chromosome. If this were to become separated, if it were to become separated, and you have something like this and something like that, separate this one. Each one of those separate. This is a chromosome, and that's a chromosome. Okay, so it's a little confusing in terms of nomenclature. If it's standing by itself, it's a chromosome. The whole structure together. This one here, this one that looks like an X, that whole thing is one chromosome, composed of two chromatids. If the chromatids separated from each other, each chromatid would become a chromosome, or be known as a chromosome. Okay, so it can be a little bit confusing in terms of nomenclature, but just think of it as the chromosome is composed of chromatids, and each chromatid can be its own independent chromosome. All right, and they usually have a short structure, a short and longer structure, a short arm, which is we call the P arm, and a Q arm. Okay. There's a center portion that is really, really pinched off. It's really constricted, and we call it the centromere. So that's going to be one of those words today, centromere. Centromere has some proteins there that allow it to attach to other chromatids. So here's the centromere. Let me draw that for you guys. Put it in red. Here's the centromere, and here's the centromere also. And there are structures, proteins within it that allow them to attach. Okay, before they separate, they're attached at the centromere, and they allow for other things to attach there as well, like microtubules that allow for the movement of the chromosomes from one place in the cell to the other. All right. Let's see. Let's take a look at this next slide. feel like I'm forgetting something. No. Okay. Okay. Oh, here's one thing I want to point out to you guys. This chromatid is an identical replica of this other chromatid. So the two chromatids are identical to each other. They both have all of the same components. So we call them sister chromatids. The reason that they are like this is because in preparation for cell division, you must replicate your genetic information in order to be able to distribute it to the daughter cells that result at the end of cell division. All right, so that's why the sister chromatids are genetically identical. There's nothing that one sister chromatid has that the other one doesn't. All right, and that's why when they separate, they're full chromosomes. Okay, so that takes us into when does the cell divide? Well, we have to think about the cell life cycle, the life cycle of the cell. All right, so the life cycle of the cell is determined by the genetic code, as is everything the cell does is determined by the genetic code that it contains. And however, all cells undergo more or less this whole cell cycle that you see here presented. All right, so the first portion will start off when the cell is being just a regular cell and doing its cell thing. And we call this interphase. During interphase, it's just doing what it's supposed to be doing, living life and creating proteins. All right, and interphase, the first part of interphase is where also that happens. We call that gap one, G1. And the cell can hang out and be like this. For example, a neuron, a nerve cell, right? It can stay at this position for many, many years. And we call that a resting stage, G naught. All right, so the cell is not dividing. It's just undergoing its day-to-day processes, living, creating proteins, and doing whatever else it was designed to do. Passing nerve impulses if we're talking about nerve cells. However, there are times when that particular cell wants to undergo cell division. And when it does so, while it's still an interphase, there is a portion of its life cycle where it will start doing things that prepare it for cell division, for nuclear division as well. And preparation for nuclear division and the division of the nucleus happens in S phase, because in S phase we have multiplication, replication of the genetic code of the genetic material, DNA. Okay, and then it'll undergo gap 2, where the cell starts to replicate all the other organelles. All the organelles, not the other organelles, but all the organelles within the cytoplasm. And it has to grow them and replicate them enough so that when the cell undergoes division, it can distribute them equally into two daughter cells, and both daughter cells will have enough. The rest of the cell cycle is composed of mitosis, which we'll talk about in a few slides, and cytokinesis. Okay, so here's the cell cycle again. Okay, let's start, for example, at the beginning of interphase, the cell is just doing what that cell is designed to do, right? And it's just growing. growing, or at some point it may stop growing, and it's continuously making organelles, replacing old organelles, making new proteins, etc. And if it stays there for a very long time, it basically enters this gap naught or gap zero cycle. Basically, it's not leaving. What it means is it's not leaving this first portion of interphase. When the cell is preparing for its cell division, it starts replicating DNA and growing other things. So that's S phase. And gap 2 is it finishes growing off organelles and enough material for it to distribute to two daughter cells. So all of that is interphase. And then it'll enter mitosis. Mitosis being division of the nucleus, which has several portions to it: prophase, metaphase, anaphase, telophase. And some of those phases overlap with cytokinesis, which is actual cell division. Okay, so let's go over mitosis. So specifically, mitosis is the division of the nucleus. And you have to divide the nucleus so that the genetic material within the original cell can be distributed equally to the two new daughter cells that result at the end of mitosis and cytokinesis. All right, and nuclear division starts before cellular division occurs. So there is a difference. Mitosis, colloquially, when you say, "Oh, you know what mitosis is," to somebody on the street, either they don't know or they're going to say, "Isn't that cell division?" And yeah, they're right. It is cell division. It will specifically part of cell division, and what part of it is that it's the division of the nucleus just before the cell divides. And cytokinesis is what is specifically referring to the division of the entire cell. Okay. Okay, so this is some of what I've already said. You guys can read through this. The S phase of the cell cycle is when the cell decides to divide, right? And it has to replicate D. And then I talked about G2 phase. I already spoke about that. Okay. So why is this important? Why does mitosis matter to us? When does it occur? Mitosis, you have to understand, mitosis is a form of asexual reproduction. That is, reproduction of the cell without sex. There's no genetic interchange or exchange or creation of new genetic material. All right, and it is used for growth and repair. You skin your knee, how are you going to heal that? You're going to heal that through the process of mitotic cell division. Okay, cell division that has nothing to do with sex. Mitotic cell division. You want to grow? Let's say you have a child, or you are a child, or you're growing some plants, some veggies, because you don't want to go to the supermarket in these times of COVID. Well, then all that growth occurs because of mitosis, mitotic cell division. So let's concentrate here on the results at the end of mitotic cell division. All right, at the end of mitotic cell division, you have two new cells that arose from the original parent cell. So you have two daughter cells. Those are the new cells, the daughter cells. And, they came from one parent cell. So that parent cell underwent mitotic cell division, and now you have two new ones. And the two daughter cells are genetically identical to the original cell, and they're genetically identical to each other. So this is like cloning. All right. So, for example, All those orchids that they sell or used to sell at the supermarkets, Trader Joe's, at Stop and Shop, at Home Depot. A lot of all those orchids, those cheap ones, $20, $17 ones, they're clones. And how do they get them? Well, they take a tissue sample from an original plant and they put it in a specific medium in viral conditions. And what happens? Well, those couple of cells in that tissue sample undergo mitotic cellular division. They're treated with some special chemicals that make them grow a little bit faster than they would out in nature. And before you know it, you have hundreds, if not thousands, of copies of the original plant, and they're all genetically identical. Right, and that's because no sexual cell division has occurred. It's asexual, and that's mitotic cell division. Right. So because they're genetically identical, the daughter cells are genetically identical to each other and to the parents. They have the same number of chromosomes as the parent cell, so there's no reduction of genetic material. It stays the same. So how does mitosis actually happen? Well, that's a little bit more complicated than everything else I've already said. All right. So there's a mitotic spindle that's involved. When the cell is getting ready for cell division, a mitotic spindle starts to grow throughout the cell, and it's composed of little proteins, microtubules, which are composed of proteins, and they are going to originate at poles, so at opposite ends of the cell. There's a little aster of microtubules that starts to grow, and by aster I mean like a star shape. The microtubules grow, and eventually they connect at the centromere, and the specific location is the kinetochore. They attach to the centromere of the chromosomes. Okay. So the mitotic spindle will originate at the poles in a specific location, and that's called the centrosome. Okay, so you have the centrosome. If we're talking about an animal cell, then you'll also find centrioles at the centrosome. Okay. So three very similar vocabulary words here. You have centrosome, centrioles, centromeres. Okay, so at the centrosome, there are some organelles called centrioles if you're an animal cell. And the centrioles play a part in the creation and growth of these microtubules, this mitotic spindle. The mitotic spindle grows from the centrosome with the aid of the centrioles if you're an animal cell. If it's a plant cell, then it doesn't really need the centrioles for some reason. And it'll grow from the centrosome and attach itself at the centromeres of the chromosomes, specifically at a set of proteins at the kinetochore. All right. So the point of the microtubules, though, is for the chromosomes to know how and where to move when it's time for them to move around during mitosis. So the stages of mitosis are for the purposes of this lab, we're only going to consider them as 4 stages. All right. So a very simplified version of mitotic nuclear division. And all you really should know are these 4 stages. Okay. And you can help yourselves with this little mnemonic: PMAT, prophase, metaphase, anaphase, telophase. Okay. So here we some pictures of prophase, metaphase, anaphase, telophase. What happens during prophase? Well, during prophase, the nuclear envelope or the nuclear membrane that surrounds the nucleus begins to enlarge and disintegrate. And I forgot to write, the chromosomes are fully condensed by this point. Okay. So by the end of prophase, the chromosomes are condensed into their chromatid form, the chromosome form. The nuclear envelope disintegrates, and the reason that happens is so that the chromosomes can move around. All right. And the next stage is metaphase. During metaphase, the chromosomes will line up at the equator of the cell. They do that with the aid of the microtubules. The microtubules have attached to them, and they're directing them to line up in the middle of the cell. And that's for preparation for the next stage, which is anaphase. Anaphase is when the chromosomes separate. So the sister chromatids are separating from each other. Remember, the sister chromatids are the two portions of the chromosome that have identical genetic material. And they were fully attached thanks to replication that happened at the end of, in S phase. Okay, so the DNA in S phase of interphase was replicated, and so when it became condensed in prophase, it appears as a pair of sister chromatids, which in metaphase they line up at the cell equator, and in anaphase, the sister chromatids separate. Okay. At the end of anaphase, cytokinesis starts to happen, and then telophase. During telophase, cytokinesis is still occurring. But during telophase, the sister chromatids are now at their corresponding parts of the cell, at opposite ends of the cell. And a new nuclear membrane or envelope is created around those. So that in essence, you end up with two nuclei in one cell. And the cell is undergoing division as well. So by the end of telophase, the cell is finishing up dividing. All right, so if we were looking at this under the microscope, this is kind of what it would look like. All right, these are pictures directly from your lab book. You have a cell with a very large nucleus and some dense structures inside the nucleus. Those are the chromosomes. Then, in metaphase, the chromosomes are lining up in the center, and at either end you have these asters, which are the microtubules forming. All right, the microtubules have formed actually, and they're connecting to the chromosomes. Then, during anaphase, the chromosomes are separating from one another. So there's a gap here now you can see, because a whole bunch of chromosomes are moving in this direction. The other set are moving in the opposite. All right, and at some point during the end of anaphase and the beginning of telophase, the cell that begins to divide, that's cytokinesis, and so you get this pinching effect in animal cells, right, which looks like the cells are about to be pinched in the center so they can be separated from one another. All right, so in cytokinesis, both daughter cells receive an equal share or a good enough share of the organelles that were duplicated during G2, gap 2 of interphase. Cytokinesis begins in anaphase and continues throughout telophase, and I do believe that it finishes right afterwards. Right, it ends by the beginning of the next interphase. Right, so at the end of telophase, that's when you have the next interphase. By definition, once cytokinesis finishes, the cell has entered. interphase. All right. So, like I was saying before, if it's an animal cell, the way that the cell divides physically, that is by the formation of a cleavage furrow. So that's this here being presented. Little pinching effect of the cell in the center. And as this pinching effect gets tighter and tighter, which is directed by a special protein that is like acts like a noose and tightens this until it breaks the two cells apart. All right, so here's a scanning electron microscope image of this. In plant cells, cytokinesis occurs a little bit differently. Why? Because there's a cell wall which prevents the pinching of the cell. The cell wall is hard and rigid and it's not easily broken down. So, um, what can happen though is the cell wall can grow. It can more material can be added to a cell wall, and then a new cell wall can be created to separate the cell itself, the new daughter cells. Okay, so the new membrane, a new cell membrane, and the new cell wall is created from vesicles that come from the Golgi apparatus. All right, and this line of vesicles is called a cell plate. All right, so here's a drawing of it. Here you have a cell that now has two nuclei because of nuclear division, mitosis. And so a cell plate starts to form and it creates a thick cell plate and eventually becomes a new cell wall along with a new cell membrane. Right. So in animal cells, you have the formation of the cleavage furrow, and in plant cells, you have the formation of a cell plate. Okay. So let's actually go to the chapter in the book. Okay, so here is chapter 7, mitosis. What's that? All right, so this is cellular asexual reproduction. All right. In this page, they're asking you to state the events of each stage of the cell cycle. So I went over them in the recitation. So yes, please do fill this in, fill it in, fill that in, fill that in, and fill that in. Explain why the entire process is called the cell cycle. Well, that's because this is, it's a cycle. At the end of mitosis, you start interphase. And at the end of interphase, you undergo cell division to start again, right? Say all sorts of things. So a whole different way, a whole bunch of different ways in which you can explain that. All right. You can say it begins with phase, ends with mitosis, and begins again. All right, so yeah, fill this in, fill that in, fill that in, and fill that in. So here we have, on this page, they're going to ask you one question on, and two fill-ins on the chromosome itself. So here it says, it is customary to call the cell that divides the parent cell and the resulting two the daughter cells. If a parent cell undergoing mitosis has 18 chromosomes, each daughter will have blank number of chromosomes. So remember, mitosis is cell division that does not experience a reduction of genetic material. So it will have the same number of chromosomes. So we'll have 18, or you can say the same. Same. Right. So here we have the centromere, and here we have sister chromatids. The process requires several phases. The process of mitosis. It says consult figure 7.2 and write the phases of mitosis, right? Remember, PMAT. So that's prophase, and then metaphase. Meta meaning middle, right? It's easy to remember because the middle metaphase, the chromosomes line up in the middle. Then you have anaphase, where the chromosomes separate, and then telophase. Telo meaning just the end. All right. Okay, so in this portion of the chapter, they're asking you to actually observe a cell, an animal cell, during mitosis. If we were in lab, of course, we'd be able to do that, and we would do that with some embryonic cells, fish. And why embryonic cells? Because when an animal is still in the embryo stages of development, it's constantly undergoing mitosis. So it's one of the best places to go looking for a cell that's undergoing mitosis. Okay. So we cannot answer how many chromosomes there are in the cell because we don't know. We don't have it with us. Let's see. Examine the phases of mitosis as depicted by the models. There would have been models in the lab. Do these models show the spindle, which is illustrated? The answer would be yes, they always do. Name two ways you would be able to recognize animal cell mitosis. All right, so there would be cleavage, the formation of a cleavage furrow, right? At the end. And another way would be the presence of centrioles. Okay, what is the appearance of the spindle pole? They're talking about the appearance of a star-shaped structure, which is the aster. Right. On this page, we would actually have been looking at some of these things under the microscope. Right. The prophase cell in figure 7.5, which is this one, so we're looking at this, has the same number of chromosomes as the telophase nuclei, this one. Explain the different appearance of the chromosomes. All right, why do they look different? Why here they look a little bit more like a single line, rather here they look like a little X. All right, so in this one they look like a little X, and here they look like a little bit of a single line. Why? Because of what happened in anaphase. So you can say during anaphase, the sister chromatids separated. In other words, the chromosomes are unreplicated at this point. So don't worry about this. This is more for write down notes. You can write down notes of your own liking there if you want, but there's no question there to answer. Okay, so in animal cells, a cleavage furrow. Here I'm over here. In animal cells, a cleavage furrow, an indentation of the membrane between the daughter nuclei, begins as anaphase draws to a close. The cleavage furrow deepens as a band of actin filaments, called the contractile ring, slowly constricts the cell, forming two daughter cells. Are any of the cells in your whitefish blastula slide undergoing cytokinesis? So if we were looking at it under the microscope, the answer is yes, but there would be very, very few of them. So you'd be looking at a tissue that is composed of many, many cells, and most of the cells would be in prophase. Many of them would be in metaphase. Some of them would be in anaphase, and a few people in the lab would be able to find something in cytokinesis. So you can see, yes, just a few. It's fine. And if you were able to see that, then you should be able to see cleavage furrows. All right. So here in this page, to talk about cytokinesis in plant cells. Okay. What is the number of chromosomes in each of the cells in your model series? Well, you can use this thing to count, maybe. Let's ignore that because these drawings are not consistent. All right. But suffice to say that they're just trying to hammer home the fact that the number of chromosomes in the beginning of mitosis is the same as the number of chromosomes at the end of mitosis. Okay, so the best way to find mitosis, cells undergoing mitotic cell division, in plants would be looking at the tip of a root. Onion roots are very easy for that. Why the tip of a root? Because the root tips are a location where plants will grow. Plants don't undergo mitosis in most of the I guess body, for lack of a better word. Plants undergo mitosis, or in other words, grow at certain places, and one of those places is the end of a root. Okay. All right. We can ignore this since we would have to actually be looking at some cells under the microscope to see this. All right, we can ignore this. Okay, so we don't have to do this page. This page you can select to just cross it out. Okay. Okay, let's see what's going on here. After mitosis, the cytoplasm divides by cytokinesis. In plant cells, membrane vesicles derived from Golgi apparatus migrate to the center to form that cell plate. All right, and here what they're asking is, in your own observations of the onion root tip, were any of the cells of the onion root tip slide undergoing cytokinesis? Okay. All right. So the answer would be, again, yes, like we did for the animal cell, but not very common. All right, not common. And how do you know? There was a cell plate forming. Where is it forming? It's forming between the two new nuclei. Between two nuclei. I ran out of space. You guys can write bigger or type in bigger, smaller, or whatever to make it fit. All right. Offer an explanation for why figure 7.8 is so detailed. All right. So obviously this is a transmission electron microscope. The image was taken by a transmission electron microscope. Right? It's a 2D image with lots and lots of details. All right, with great resolution. What would you predict that the vesicles of the cell plate lay down the new cell wall inside or outside the vesicles? Okay. So you guys can think about that. All right. You can do. I'm not going to hold you responsible for this one. So, I'll write it here. Not responsible. Okay. All right. And that brings us to this chapter. This page. We did this page, and that brings us to this page. So, first question. What is the name for the period of time when a cell is performing its usual functions and replicating DNA? All right, specifically, when it's replicating DNA. That's S phase. S phase, and that's in intraphase. When does nuclear division occur? Well, nuclear division occurs only during mitosis. In terms of this chapter. So there's also meiosis, which we're going to cover next. Why do organisms perform mitosis? What's the purpose of mitosis? Remember, it's growth and repair. List the 4 stages of the cell cycle. All right, so you guys can write it out. PMAT, right? I'm going to write PMAT. You guys can write it out. Prophase, metaphase, anaphase, telophase. In that order. What occurs during the S stage of interphase? DNA replication. That's kind of a repeat question there. What is the term used to describe duplicated chromosomes? In that case, we call them sister chromatids. We're talking about this is being a chromosome, and here's another one, right? So these are sister chromatids. The whole thing is a chromosome. Okay. If asters, you're talking about the microtubule formations at the poles of the cell, are observed in cells undergoing mitosis, are the cells animal or plant cells? All right, so the asters are only visible for animal cells. And that's directly to do with the presence of centrioles. What structure that helps chromosomes move forms during prophase? You can just simple and say mitotic spindle. When do chromosomes align at the equator of the spindle? And that would be in metaphase. What happens to sister chromatids during anaphase of mitosis? They separate. During which phase of mitosis does the nuclear envelope reform and the nucleolus reappear? And that would be, the nucleolus is the inner portion of the nucleus. That would be during mitosis. So that would be at telophase, the last phase of mitosis. When are organelles divided up between daughter cells? And that's during cytokinesis. Cytokinesis. Cyto meaning cell and kinesis meaning separation. In which phase of the cell cycle do cells spend most of the time? Okay, most of the time is spent in G1, or say gap 1. What structure gives rise to new plant cell walls? Okay, and that would be the cell. Why is it necessary for chromosomes to duplicate before mitosis? You can say something simple, like for the equal distribution, right? The equal distribution of genetic material in the resulting daughter cells. Why is it important for sister chromatids to be attached to each other during the beginning phases of mitosis? All right. Again, this has to do with distribution. All right. If they're not attached, then how would you know? How would the cell be able to control whether one cell got too many copies or too few? All right. So this is you can just say to maintain. I don't like the way that handwriting came out, so I forgot one word. Apologies. To maintain the correct number of copies, right, of the chromosomes in the resulting daughter cells. Okay, list several parts of your body where you would expect to find actively dividing cells and several parts where you would find mostly resting cells. All right, so we can make a little chart. Resting cells and dividing cells. Oops. Well, I'll tell you one thing, guys. Nerve cells, right? Nerve cells are usually resting cells. I'm not sure about bone cells. Bone cells are not actively being replaced all the time. I guess we could put it down. Bone cells, right? Muscle cells, they do get replaced. Bones and muscle cells do get replaced, but only when some sort of damage has occurred. Nerve cells are a little bit harder to replace, even after damage. So bone cells, muscle cells, maybe. You guys can look it up. Actively dividing cells, what's a good place for that? Well, cells that get used up quickly. For example, the lining of your inner cheek. Okay, so those cells get replaced very quickly. So you can say cells lining your inner cheek. Cells that are lining your stomach and intestines. These are cells that get scraped off very easily with the food and the refuse that's passing through, and they have to be continuously placed. Skin cells also. All dead skin cells are always being flipped off, and you need replacing. And look, whenever you get hurt or something, you need to heal the wound with mitotic cell division. Now let's get back to the PowerPoint. So at this point, we're now going to talk about meiosis. Yeah. Okay. So meiosis, the purpose of meiotic cell division is for the creation of gametes. The creation of gametes is known as gametogenesis. And what are we talking about when we say gametes? We're talking about sperm cells, egg cells, sperm cells for males, egg cells for females. And of course, meiosis therefore only occurs in the organs of the body that create these cells, the gonads. We're talking about the testicles. We're talking about the ovaries. Meiosis is different from mitosis in several ways. Number one, mitosis creates 2 daughter cells. Meiosis creates 4 daughter cells. Mitosis creates genetically identical cells without reduction of the amount of genetic material. But meiosis, you do reduce the amount of genetic material. So we're talking about 4 haploid daughter cells. Haploid meaning they only have one set of chromosomes. Keep that in mind. So in order to go from the original cell, which has 2 sets of chromosomes, to 4 daughter cells that has one set of chromosomes, you've basically taken out half of the cells, half of the genetic material. So we reduce the chromosome count by half, and the daughter cells are genetically distinct from each other and from the original parent cell because genetic recombination does occur. All right. So there are some terms we have to know about homologous chromosomes. All right. Just like in mitosis, the chromosomes before meiosis occurs are replicated. So you end up with the sister chromatids. You end up with at the end when you see them in mitosis. But remember, um, most all animals and all plants and fungi that undergo sexual reproduction inherit a set of chromosomes from each parent. Okay, so there's a set of chromosomes from the mother and the father that the offspring, the children, or the progeny, have inherited from each one of those parents. And when a cell undergoes mitosis, all the chromosomes are doing their thing independent of each other. But in meiosis, they are not all doing their own thing, all these chromosomes. So what happens is, unlike in mitosis, in meiosis, homologous chromosomes pair up. So what do we mean by homologous chromosomes? So, like I was telling you, you inherit one set of chromosomes from one parent and the other set from the other parent. But the two sets carry the same kinds of genes. So chromosome number 1 from the mother carries the same genes that chromosome number 1 from the father did. And so on. Depending on the species, depends on how many chromosomes. So homologous chromosomes, like chromosome number 1 from the mom and chromosome number 1 from the father, are homologous chromosomes. And when they're in your cells, they're doing, when they condense into chromosomes, they pair up when they're going to go through meiosis. When it's mitosis, they don't have anything to do with one another. They just want to replicate properly. But in meiosis, they pair and for a very specific reason. Okay, so remember, homologous chromosomes, these are different chromosomes one from each parent, but they carry the same types of genes. All right, and when you're an animal or a plant or some sort of being that has two sets of chromosomes, then you're considered to be a diploid individual. A regular cell in the rest of your body, in almost all of your body, will be a diploid cell. And animals, plants, fungi, whatever, that contain two sets of chromosomes are also considered diploid. However, there are some living things out there that only one set of chromosomes, and your sperm cell and your egg cells will be haploid. That's the result of meiosis. And haploid meaning they only carry one set of chromosomes. Okay, so let's move on here. Why do the homologous chromosomes pair up? All right, so the homologous chromosomes pair up for a very important reason, and that's for genetic exchange. All right, so at the beginning of meiosis 1, one of the other differences between meiosis and mitosis is that meiosis occurs with 2 rounds of division, not just one. Okay, so at the beginning of meiosis, in the first round, we have prophase, metaphase, anaphase, telophase. And we call it prophase 1, metaphase 1, anaphase 1, telophase 1. And at the beginning of prophase 1 is very similar to mitosis. The nuclear envelope disintegrates, the chromosomes are condensed. The difference is the chromosomes pair up into homologs, right? Homologous chromosomes pair up, form these things called tetrads. kind of looks like, here's a set of sister chromatids. Let's see, this blue one here is from one of the chromosomes that you inherited from your father. And let's say this pink one here is from your mother. Now, this is a pairing of two homologous chromosomes. This is one, and this is the other. Okay, and they pair up right here. They connect at the kinetochore or at the centromere. And what happens? Well, they undergo synapsis, crossing over event. In essence, the homologous chromosomes exchange genetic material or parts of the chromosomes. That's important. Okay. So this whole thing is now called a tetrad. Right. So now you have the problem of separating the tetrads, which is why you have to undergo two divisions. So in metaphase, the tetrads line up in the cell equator, and in anaphase 1, the tetrads, not the sister chromatids, the tetrads, become separated from one another and move to opposite poles of the parent cell. This is very much like mitosis, except it's the tetrads that separate, not the sister chromatids. And then, of course, phase 1 is the opposite of prophase 1. The nuclear envelope forms around the separated chromosomes at poles of each parent cell. The kinesis begins. All right, so now each pole has one chromosome from each pair of homologs. So it has one set of chromosomes. And I may have forgotten to write this. Okay, so what this means is at the end of meiosis 1, haploid daughter cells are created. So at the end of the first round of meiosis, the cells go from being diploid, having two sets of chromosomes, to being haploid, having one set of chromosomes. Okay, so here's synapsis, a closer look at the crossing over event. Here you have a tetrad, which should be attached, by the way. And the tetrad will exchange genetic material. So one chromosome, one homologous chromosome will exchange genetic material with the other. And here is a simple representation of that. They literally just, the arms of the chromosomes overlap and they exchange pieces of the chromosome. This is not something that happens only once or in certain portions or only one time in the same place. Crossing over occurs many, many, many times during prophase. So this portion gets crossed over, this portion gets exchanged, that portion with this one with that one, and then this one does it again several times, all of them. So there's lots and lots of exchange, right? And this exchange creates an endless supply of variation in genetic makeup. All right, so by the time crossing over is done, the homologous chromosomes are not the same as they were before they underwent meiosis. So the homologous chromosomes are quite different already. So the daughter cells that result not only have only one set of chromosomes, but the chromosomes themselves, the genetic material is different from the original parent cell. and from all the other resulting daughter cells. All right, so in meiosis 2, the second round of division, in this case, the situation is almost identical to what you found in mitosis. All right, and for the purposes of this lab, it's the same thing. It's identical. So remember, at the end of meiosis 1, each one of the daughter cells receives one of these chromosomes. So one daughter cell will receive this chromosome, and the other daughter cell will receive this other chromosome, which are composed of sister chromatids. You see that? This is a sister chromatid, and that's a sister chromatid. This is a sister chromatid, and so is that. All right, so okay, so in the beginning of prophase 2, of meiosis 2, the nuclear envelope disintegrates, the chromosomes are condensed again, and the sister chromatids start to attach to the microtubules. At metaphase 2, the sister chromatids line up at the center of the cell equator. In anaphase 2, the sister chromatids separate from each other, and at telophase 2, the sister chromatids are migrated to opposite ends of the cell, and a new nuclear membrane is created around each set of chromosomes. Right? And then you undergo cytokinesis again. Okay, so here's another representation of that. Let's start over here again. Oops. Okay. So it's a little hard to see, sorry about that, but here's prophase 1. Homologous chromosomes pair up and they exchange genetic material. And then in metaphase 1, the chromosomes line up in the center, then they separate. And at anaphase 1, and in telophase, these now haploid nuclei end up in new cells because of cytokinesis. At the end of meiosis 1, you have a short resting period, and then it undergoes another round of division. Right? Each one of these cells, which is a result of, these are the daughter cells from meiosis 1, will now undergo their own cell division. Right? So the chromosomes reappear and the nuclear envelope disintegrates. Then in metaphase 2, the chromosomes line up at the center of their corresponding cells. And in anaphase 2, they separate. The sister chromatids separate. And in telophase 2, you get cytokinesis, right? And the formation of new nuclei. And note, this cell is different from this cell, which is different from this cell, which is different from that cell. All 4 cells are genetically different. They all have one set of chromosomes, and so erase the word copy. They all have one set of chromosomes, and all the chromosomes are different in terms of their genetic makeup. They all have the same types of genes as all the other ones. Nobody's missing any information, but because of the crossing over that occurred, the genetic makeup is different. Right, so each one of these cells is different from each other, and it's different from any of the original cells, which are also different from any of the cells from the beginning of meiosis. All right, each one of these cells will become either an egg cell. Depends on the species. So if it's like, for example, a human, 3 of these cells will wither away, and one of them will become an egg cell. And I don't know how the decision gets made about which one survives and which ones wither away. But in males, each one of these will become a sperm cell. All right, so this brings us to the importance of sex. Why? Why is there sex? Because in the formation of gametogenesis, you have genetic recombination, which is synapsis, the crossing over event. Then you have to think about, all right, so each cell has its own copy or its own set of chromosomes, but which gamete got which set? Well, that happens randomly, and that's independent assortment. So in the end, you don't know which gamete is going to be out there fertilizing which other gamete, which sperm cell is going to fertilize which egg cell. Okay, so these newly recombined chromosomes assort independently of one another into different daughter cells. All right, so think of it this way. Um, the chromosomes. Remember, I said that you inherit one set of chromosomes from one parent and the other set of chromosomes from the other parent, and then they cross over, they exchange genetic material, and they end up separating. But what you have to remind yourself of is that the chromosomes that came from one parent don't all end up in the same daughter cell at the end of meiosis. Now all the chromosomes get scrambled. So yeah, each daughter cell gets a copy of chromosome 1, chromosome 2, 3, 4, 5, but not all those chromosomes did not originally from the same place, from the same parent. It's mixed. So the daughter cells have a mix of chromosomes from each one of the sets that the individual who has these cells that are undergoing meiosis has inherited. Okay, so think of it this way. Let me see if I can draw it. So here's a cell. Here's a chromosome. Okay, let's make a K. Maybe not that. This is the whole thing, huh? All right. So here's a chromosome, which is a homologue of something else. Here's another chromosome. So here's one set of chromosomes that was inherited from one parent. The blue one is from the other parent. Okay. Make this a little bit longer just to be able to distinguish them. All right, so this is a little bit longer. Right? They undergo genetic recombination. So now the cell looks like this. Okay. And the same thing over here. The little chromosome. Okay, so what am I showing here? I'm showing the same chromosomes which have undergone crossing over. They've undergone crossing over here and here. So this mostly red chromosome has a little of the blue genetic material, and this mostly blue chromosome has a little bit of that red material. Well, what happens next? Well, the cell under the homologs separate, right? And you end up with a cell that looks like this. You know, it's a little cleavage furrow going on here, and you have this side may have this chromosome, and okay, drew it a little bit upside down there. You get the idea. And this cell will get these chromosomes. So what am I trying to show you here? Okay, eventually, right, the cell will separate. So you end up with something. One cell that has this set of chromosomes, and the other cell that has that set of chromosomes. All right. Even though these homologous chromosomes, even these homologous chromosomes were together, this is blue. This is blue coming from, you inherited these two chromosomes from the same parent, and you inherited these two red chromosomes from another parent, the same other parent. That doesn't mean that they end up together in the same daughter cells. So in this case, one daughter cell has one of these blue chromosomes and one of these little red ones. And remember, so this is independent assortment. Remember, humans, for example, have 23 pairs of chromosomes. It's the which chromosome ends up in which daughter cell is completely random. Okay, so that's independent assortment. So you have synapsis or genetic recombination, independent assortment, and then during the act of sexual reproduction, or actually afterwards, when the gametes meet, you have random gametes fertilizing each other. So which sperm cell is it going to be that's going to fertilize the egg cell, or which egg cell? As we know, in humans, usually you only have one egg cell, but in other animals, there are animals that have hundreds of egg cells being fertilized at the same time by millions of sperm cells. Which sperm cells are going to fertilize which egg cell? It's up to chance. So the result is there are trillions of possible genotypes or genetic makeups that are possible for the creation of the next generation. So basically, all this is done just to maintain variation within the population. Okay, so now let's go to the second chapter. So we do some screen sharing. Okay. Chapter 8. So chapter 8, sexual reproduction, meiosis. Okay. In this one, talking about distinguish between diploid and haploid. It's easy enough. Diploid equals having two sets of chromosomes. Haploid having one set of chromosomes. Distinguish between a homolog and a tetrad. All right. A tetrad is when you have the two chromosomes, two separate chromosomes, joining. And which two chromosomes are joining? Well, those are the homologous chromosomes. All right, and the homologous chromosomes are the homologs. They don't have to be joined, right? The homologous chromosomes don't have to be joined. They don't join during mitosis. All right, so don't make the mistake of saying, well, cells that undergo mitosis don't have homologous chromosomes. That's not true. Of course they have homologous chromosomes. It's that the homologous chromosomes don't come together. The homologs are chromosomes that have the same what do you call it? Same types of genes. Not necessarily the same genetic makeup, just the same types of genes. And inherited from each parent. Separate parent. It's a long answer. Okay. Okay, so let's see. Okay, using figure 8.3, which was this one, as a guide, try to find the cell in each phase of meiosis, tell what is happening in cases. Okay, so you can still do this because all you have to say is describe what's happening. In prophase, the homologs, you just rewrite what you wrote, what you see written there. The homologs pair up into tetrads. In metaphase 1, the tetrads or the homologs line up at the cell equator or spindle equator, however you want to say it. In anaphase, the tetrads separate. Remember, in meiosis 1, the tetrads separate, not the sister chromatids. And in telophase 1, a new nucleus appears around each set of new chromosomes. Oh, sorry, each set of separated chromosomes or tetrads chromosomes. So basically this is a new nucleus being formed around this set of chromosomes and another one around this one. And maybe you can say cytokinesis occurs. And in this case, tell what is happening in each of these phases. Okay, prophase 2. In prophase 2, just like mitosis. Okay, nuclear envelope. Oops, didn't write that well. nuclear envelope disintegrates. Right, the nuclear envelope disintegrates, and I guess you can say the chromosomes are visible. In metaphase 2, chromosomes, just like in mitosis, chromosomes line up in the cell equator. In anaphase 2, the sister chromatids. The sister chromatids separate. And in telophase 2, new nucleus forms for each daughter cell, right? Just like in mitosis. Okay, why is it important for both sperm and egg to have the haploid number of chromosomes? Because they will receive a set of chromosomes during fertilization. All right, so if they're going to receive a set of chromosomes during fertilization, if you didn't have the haploid number, one set of chromosomes in your egg or sperm cell, then the resulting fertilized cell will have 3 sets of chromosomes, all right, or 4 sets of chromosomes. So you want to maintain it as diploid. You can say must maintain diploid condition. In the resulting zygote. The zygote is the fertilized egg. So that's why we must have haploid number. All right, on this page, they're talking about going through the steps of meiosis with the use of these little pop beads, which help you model the event of meiosis. Okay, and here you kind of do the same thing over. All right, which takes us to this page. Okay, we don't have to deal with this since we're not in the lab. Okay, so we don't have to deal with this. We can skip that. Summary production of variation during meiosis. Meiosis reduces the chromosome number. If the parent cell is 2n, that's diploid, and 2n, or diploid, means for this individual animal or plant or whatever, it means having 4 chromosomes, 2 sets. Okay, 2 sets of chromosomes, 4. The daughter cells are n, which is 2. N equals 2. 2n is equal to 4, n equals 2. During prophase 1, the homologs come together and exchange genetic material. Now, the inherited chromosomes will be different from those in the parent cell. This process is called synapsis. If you put down crossing over, that's okay also. During metaphase 1, the homologs align independently. And therefore differently. This is the second process that produces genetic variation through sexual reproduction. During fertilization, variant sperm, random sperm fertilize random eggs, further helping to ensure that the new individual inherits different genetic combinations, right? Or just genes of homologs than the parent had. Okay. Oops, too much. So that was this page. In this page, let's see, a summary of the human life cycle. Okay. In this case, they're asking name of organ that produces the gametes in males is the testicles, right? Testicles. In females, it's the ovaries. The name of the process that produces gametes in males is spermatogenesis. Spermatogenesis, and here it's oogenesis. Ooe meaning egg. Type of cell division involved in the process? Well, obviously, it's going to be meiosis, right? Same thing here. You can't get gametes from mitosis in animals. You can do it in plants, but not in animals. Name of gamete. In males, it's the sperm. In females, it's the egg. Number of chromosomes in gamete. In males, 23, right? And 23, because we're talking about humans. We can say, you can be more generalized and be half. Say half the number of chromosomes. Results of fertilization. How many chromosomes? You have 46. Number of chromosomes provided. 23 from each. You can say from each parent. Okay, so in this page is a summary of mitosis versus meiosis, which is important. Okay, so the differences between mitosis and meiosis. Okay, how many cell divisions in mitosis? We can just have one. In meiosis, 2. Chromosome numbers in daughter cells. Well, you end up with 2 chromosome numbers. Not number of cells. Chromosome number in daughter cells, the same for mitosis and half for meiosis. Number of daughter cells in mitosis, you end up with 2 daughter cells, and in meiosis, you end up with 4 daughter cells. Okay, mitosis compared with meiosis 1. In mitosis, in prophase, there's no pairing of chromosomes. Right? There's no pairing. Chromosomes do not pair at all during mitosis. In meiosis 1, prophase 1, the homologs pair. In mitosis, metaphase, duplicated chromosomes at the metaphase plate, right? So the chromosomes, they're duplicated because they have sister chromatids. They're composed of sister chromatids. And they align at the plate, at the metaphase plate or the cell equator. Metaphase 1 of meiosis, it's the homologous pairs that line up. The sister chromatids separate in anaphase of mitosis, it's the homologous. Wait, let me erase that. Homologous pairs separate. Oh, and for prophase 1, the homologs, let me erase this. Homologs check my spelling. Homologs. The homologs pair and synapsis. Okay. Telophase 1, in telophase of mitosis, chromosomes have one chromatid. In telophase 1, each daughter cell has one set of chromosomes. In other words, haploid. Okay, here they're still diploid. Okay, so now provide the correct term for each definition. Type of cell division that keeps the chromosome number the same and occurs during growth and repair, that's mitosis. Type of cell division that reduces the chromosome number and occurs during gamete formation, meiosis. Half the diploid number of chromosomes. Okay, so that's haploid. Male gamete with the n number of chromosomes, sperm cell. Female gametes with the n number of chromosomes, egg cell. So here's a comparison, mitosis and meiosis. Right. So the review questions. What are the gametes? Haploid cells, right? Haploid cells, specifically sperm and egg. What is the purpose of meiosis? Sexual reproduction. Well, no, let's not call it sexual reproduction. Let's call it gametogenesis. Right? The end game, of course, the purpose is sexual reproduction. But all that meiosis does is the production of gametes, sperm and egg. How many divisions occurred during meiosis? You have 2. Where would you expect to find meiosis taking place? In humans, in the gonads. You can say gonads, you can say ovaries, testicles. Prior to meiosis, during which stage of the cell cycle does DNA replication occur? It's the same as before, right? S phase of interphase. What are chromosomes that look alike? They look alike and carry the same genes. You know, the same genes for the same traits called homologous chromosomes. Parent cells are diploid. Daughter cells are haploid in meiosis. What term describes the pairing of chromosomes during prophase 1? The formation of tetrads. I believe that is the answer they're looking for. During anaphase 1, the homologs or homologous chromosomes separate during anaphase 2, the sister chromatids. So I ran out of space, but you guys can put it in the right. Sister chromatids. The sister chromatids separate. Which processes increase variation during meiosis? So you have three things, right? You have genetic recombination, independent assortment. I don't know why they give you such small space, such a long answer. Assortment. And I guess you could leave it at those two. The other one, of course, being something that happens way after meiosis has occurred. Because then that would have been random fertilization. But we'll skip that one. Let's keep it to what happens during meiosis. Genetic recombination and independent assortment. What is the exchange of genetic material between non-sister chromatids? That's synapsis, again, synapsis, or genetic recombination, or crossing over. During what phase does crossing over occur? That would be prophase 1. It's very important. Prophase 1, not just prophase. You have to say prophase. Crossing over does not occur in prophase 2. And there are 2 prophases in meiosis. During what phase do homologs separate? That would be anaphase 1. Again, anaphase 1, not anaphase. Anaphase 1. If a cell has 48 chromosomes, how many does each daughter cell have at the end of meiosis 2? 24. Meiosis did not reduce the chromosome number of gametes. Uh, so you don't. Let's see. Describe what would happen if meiosis did not reduce the chromosome number of gametes. Gametes, you could say. I mean, there's so many answers. The gametes could, well, not could, but would be diploid, right? And if the gametes are diploid, then the zygote, if the gametes were to fertilize another gamete, then the resulting zygote would be triploid or quadruploid. You know, they have multiple sets of chromosomes. What events occur during meiosis and fertilization that result in offspring having a mix of their parents' traits? Okay, so these are the 3 conditions. Genetic recombination, you have genetic recombination, you have independent assortment, and random fertilization, you can say. random fertilization. What are the similarities and differences between mitosis and meiosis? There are a lot of them. I'd rather not sit here and write them all out. So you guys can please fill this question out. You guys can rehash, go back to parts of the video where I describe those similarities and differences. All right, talk about how many daughter cells result, what's going on with the genetics, are they identical? So I do list them in the PowerPoint that I went over, so you guys can list them there. All right. Otherwise, that brings us to the end of this lab. Tomorrow we're going to be discussing a little bit more on genetics, and it should be a little bit more fun than this one. But you guys really do have to keep up with this lab in order to understand tomorrow's lab, all right? So make sure that you watch this one before you watch tomorrow's lab.